before we go ahead with this, couple of announcements. So the first one is for the antibiotics table that I had you guys fill out. I want to go over actually what of it do you need to know for the exam. There's more information than you need on there. And I did put up a key for this, so it's in the same lesson, Antibiotics Lesson 1. It's in the same location as the homework. It's just a separate file below the homework file itself, and it's labeled as key. So, things you need to know. The classes of antibiotics, not the specific antibiotics though. The protein targets. And then the last part that you need to know is what do the antibiotics actually inhibit. Now, there are exceptions to this. So let me walk through those particular exceptions. You do not need to know the protein targets for the following. The sulfa drugs. It is a protein known as dihydropteroate synthetase. No purpose in knowing what it is because we will never talk about it. So that's one. Sulfa drugs, the SUL. I think the name, like the one drug is sulfanilamide. So the sulfa drugs are a class of drugs.
You don't need to know what for that?
You don't need to know the protein target. You do not need to know the protein target for isoniazid or ethambutol because those are confusing. Isoniazid and ethambutol, they're grouped together on the table. And the last one that you don't need to know is the target for or the specific protein target for bacitracin. The rest of them, you do. Because they are things that you have honestly, with the exception of the penicillin binding proteins, which we'll talk about more a little bit today, but more so on Friday, you have seen the rest of those in some way, shape, or form, either in this class or others. Danica.
Do we still know like the process inhibited for each of them?
You should know the process inhibited for each of them. Yes, yes, and we'll cover the isoniazid and ethambutol today. We covered bacitracin on Monday as saying that it just inhibits protein, or it inhibits cell wall synthesis, and we'll cover the sulfa drugs today as well. Allie.
Are these like from the chart or like the mini lecture?
These are from the chart that we worked on for Monday's class. I'm not asking you to know about the spectrum because, to be completely honest, for example, the beta-lactam antibiotics, there are some that are narrow spectrum, there are some that are large spectrum. And so it's kind of pointless to say memorize all of them, what the spectrum is. Instead, what I'd be asking you to do in terms of the exam, at least for this one that's coming up, is to say, for example, if I said, ampicillin targets gram-positives and gram-negatives. Therefore, by definition, it is a broad or narrow-spectrum antibiotic. And you would give the appropriate answer given that it targets gram-positives and gram-negatives. Okay. Later on, and I don't think this is something we're going to get to until the next unit, is talking about how do you choose? How do you choose on the best antibiotic? And that's something we'll save a conversation for later. Any questions on those? Apparently we've got people watching the class from outside, and it's making me nervous. Avoid eye contact. All right. They're students. They're people. So look happy. Look happy and excited that you're here.
Run!
Laugh real loud. Anyway. All right. Stop it. Stop it. All right. So that was comment 1. Comment 2 was review session tomorrow. So, half the class said 4 to 6, other half said 5 to 7. So what am I going to do? I'll be here from 4 to 7. So, Here's how I'm going to do it though. So I emailed registrar and said, please set aside 108. So plan to meet in 108 if we have what we call the therapeutic index. So if you'll remember, I think we defined that the therapeutic index is really just a ratio. And that ratio is, so TI is equal to the toxic dose divided by the therapeutic dose. And these are defined in laboratory settings. Sometimes they might be defined in animal mouse settings or ultimately clinical trial settings. So the toxic dose, if you'll remember, this describes to us how much does it take to be toxic to an individual. Typically these numbers are based off of a study population, and usually the amount is either the average or the median concentration that caused 50% of the people to feel toxic symptoms. Nausea, heartburn, indigestion, upset stomach, diarrhea, hey, Pepto-Bismol. All right, so that's one. Therapeutic dose, on the other hand, is how much does it take to cure the infection? And oftentimes these are based off of things like the minimal inhibitory concentration. There are other features that go into it. How fast does the human body metabolize the drug, for example? Okay. What we are looking for are large therapeutic indices, meaning high toxic dose, takes a lot of it to cause illness, and low therapeutic dose, doesn't take a lot to cure the infection. So we're looking for large values there. Remember also, or recall that one of the things we said was that the toxic dose is inversely correlated to how toxic do we consider it to be. Something that is a very toxic drug will have a low toxic dose, low toxic dose. That's what I said. That's what I meant. Let me rephrase that because I screwed myself up. Something that's toxic has a low toxic dose. High toxicity, in other words. Okay, good, no problem. So, polymyxin B is an antibiotic that is only used topically. It's only used for triple antibiotic ointment. And that's not because it just targets gram-negatives. Actually, that's kind of a benefit to it as a drug. It's not only that it targets the outer membrane, that those two are inclusive of each other. It's not that it's selectively toxic, and we'll come back to that in a minute. It's actually because it has, no, it's not D. It doesn't have a high therapeutic index. It's not selectively toxic. That's what I'm trying to say. Sorry, apparently I'm not on top of my game right now. A high therapeutic index would be really good. It would mean that this drug is not toxic. But what we oftentimes find is that if a drug is toxic, in part it's toxic because it's not only targeting bacteria, it's targeting our own cells as well. And so we use this term sometimes, selective toxicity, to refer to the mechanism of antibiotics. Let's do an example real quick. Penicillin is part of the beta-lactams. What do the beta-lactams target? So what, not the particular protein, but what process do they inhibit? Cell wall synthesis. The particular protein that it targets is known as penicillin binding protein. There's numerous ones of it. Do our cells do we have cell walls?
No.
No, thank you, somebody, for speaking up and answering that question. I was frightened. We don't. We don't have penicillin binding proteins. Therefore, penicillin is highly selective. Doesn't target our cells, targets exclusively bacterial cells. Polymyxin B, on the other hand, targets membranes. Do our cells have membranes? Sure as heck they do. Bacterial cells as do. What it ends up doing is increasing the permeability of those membranes. So for those reasons, we consider polymyxin B to be less selectively toxic, and therefore the toxic dose, oftentimes we find with these less selectively toxic drugs, is lower. Okay? Now, think about other examples. So aminoglycosides and chloramphenicol, what do they target? Translation. What machinery is required for translation, Abby? Yeah, or what protein is required? Ribosome. Do we have ribosomes? Why doesn't chloramphenicol cause us to get sick? And it comes down to the structure. Sure, we have ribosomes. We have DNA gyrase, which is targeted by the quinolones. We also have RNA polymerase, which is targeted by the rifampins. But what's different is the structure of our version of those proteins is distinct enough that the antibiotics can't bind and inhibit them in the same way that they can to bacterial cells. So our ribosomes are, they look similar, but they're distinct enough that they don't actually get targeted by those antibiotics. That's another example of selective toxicity. Any questions on that concept or the concept of the therapeutic index?
So just to class and distinguish between bacteria cell beside, like, instead of ours just regular.
Yeah, and in distinguishing, it's not like it can make a decision, right? It's a chemical. It will, just like a competitive inhibitor in some ways, it binds to the bacterial versions of the proteins. It doesn't fit into the active site of our versions of the proteins. Would probably be one way to describe it. All right. Let's do, no, discard. All right. Which of these is a narrow spectrum antibiotic that inhibits the synthesis of mycolic acid? So A, isoniazid, B, rifampin, C, fluoroquinolone, D, streptomycin. Take a second to think about this one. Use your table if you need to. Get familiar with the information. Before we even answer this one, where do we find mycolic acid? What type of bacteria is this associated with? Yeah, acid-fast bacteria. Prototypical member, Mycobacterium tuberculosis. So one of these drugs, actually two of them, are used oftentimes to treat Mycobacterium infections. So we're finding Mycobacterium are gaining resistance to these combinations. Which of these targets mycolic acid synthesis? Yeah, it's the isoniazid. Now, anybody remember the quality of mycolic acid? We kind of described it as having a certain quality to it. Yeah, it's waxy. And typically that waxy coat, it inhibits the ability of macrophages to engulf these mycobacterium and digest them or break them down. When treating with isoniazid, prevents the mycolic acid from being deposited on the outside, gives our macrophages a leg up, essentially, in killing these cells and destroying the infection, essentially. The other drug that's often used to treat mycobacterium infections, rifampin, which, anybody know what this one inhibits? I think we just covered that. Yeah, this is our RNA polymerase inhibitor. So those two are oftentimes used together. Isoniazid is incredibly rare, narrow spectrum because it targets something that is unique to those acid-fast bacteria and not something we find in other types. All right, all beta-lactams are narrow spectrum, inhibit cell membrane synthesis, have a common ring structure, or are non-competitive inhibitors. A, B, C, or D. Oh, isoniazid. I'm sorry. Isoniazid was the correct answer. All right. So what do all beta-lactams have in common? So I hear ring. I hear, what else did I hear? B. Inhibits cell membrane synthesis. Okay. Oh, somebody picked up with that. Yeah. What do beta-lactams inhibit? Cell wall, not cell membrane. So that's that's one key distinguisher there. So it actually is C. Let's go and take a look at this structure real quick. Now, beta-lactams are a really large class, and they have been subdivided into other classes. All beta-lactams have this structure right here. And this is the ring that we're talking about. So it's 3 carbons, a nitrogen, and a double bonded oxygen. This is known as the beta-lactam ring. And whether you have penicillins, which have a 5-membered ring attached to it, or cephalosporins, which have a 6-membered ring attached to it, or monobactams, which is just this ring, or carbapenems, which are different. They all have that ring in common. In other words, they all target the same set of proteins. And all of them are competitive inhibitors. Meaning what? What's true of a competitive inhibitor? They get in the active site of their target, or what they're going to inhibit. And I mean, it did not take a genius to name this one, but these are, the proteins they bind to are known as penicillin binding proteins. And they were identified for their ability to bind to that antibiotic. So before folks knew what the function of this enzyme even was, they knew that they had an antibiotic to block it. What they found out after the fact was penicillin binding proteins help to build the cell wall. In particular, they don't do anything to nag and NAM. They actually take those tetrapeptides that connect the separate polysaccharides together and they bond them. They create a covalent bond. The one that they typically create is between 2 alanine molecules, so this is that structure that appears. Look at the similarity between that and what's true about competitive inhibitors. They look like what? They look like the substrate. Here's our substrate. There's penicillin. And one of the commonalities is this kink structure right here, this nitrogen located here, a carbon located over here, carbon-oxygen double bond. Those are all features within this beta-lactam ring. Right. Now, one thing that is unique about penicillin, or the penicillin and its derivatives, the other beta-lactams, they bind irreversibly. In other words, they create a covalent bond actually with the protein when they compete for that active site, and now that protein is completely useless. The only way to get rid of the penicillin at that point is to destroy the protein and start all over. What's this do? How do these cells die? So what does this do to the cell wall structure? It makes it more permeable, it does. Weakens it. How do these cells likely die? Exactly. And there are these great pictures of cells being treated with ampicillin, and basically what you've got is a bacterial cell that, if it's a rod-shaped one, it's basically kind of like blown out a little bubble, kind of like how a balloon pops. If you were to capture that image of a balloon popping and how it's exploding out at one end, that's what happens to these cells. That cell wall becomes so weak that it can't retain that turgor pressure that gets built up in the cell, and so these cells basically burst. Go ahead, Zach.
So what do the penicillin binding proteins do? I mean, they work with penicillin, you're saying?
So the penicillin binding proteins, their main function is to take these like long peptide strands that form in between the polysaccharides and the peptidoglycan, they covalently bond them together. And we talked about how cell walls have basically 2 forms of strength. You've got the polysaccharide, which strengthens it in the lateral direction, and then in the top to bottom direction, you've got these peptide bonds, which help to hold it together as well. So essentially, penicillin binding proteins are helping to create this lattice or this matrix-like structure that is the peptidoglycan. When penicillin binds, it prevents the penicillin binding protein from even creating these bonds. And so that then kind of doesn't allow for that longitudinal strength to be created. It only still allows for the lateral strength. So it kind of weakens up that meshwork essentially. Okay, we'll come back to this. We'll recover this a little bit on Friday because we're going to talk about actually another class of antibiotics that inhibit the penicillin from functioning. All right, next one. Rifampin specifically inhibits which step of the central dogma? Kind of already covered this, but not a bad one to recap. Yeah, it's inhibiting RNA polymerase, it's inhibiting transcription. So one of the things that I want to point out here real quick is that you guys did those concept maps. We're going to come back to that. What you need to know for the exam really are three things. Translation is controlled by the ribosome. Transcription involves the RNA polymerase. And replication involves the gyrase in some ways. And the reason why I want you guys to know those three particularly, they're all targets of antibiotics. Okay? They're all targets of antibiotics that we talk about.
Can you repeat them?
Yeah. Gyrase is involved in replication. The RNA polymerase is involved in transcription. And ribosomes involved in translation. And so we'll come back to this next week. We'll actually kind of recap those concept maps and add some deeper levels on Wednesday. But for the exam, that's what you need to know. All right, last, sulfa drugs, or sulfonamides, as they are fully known. A are competitive inhibitors, B block synthesis of folic acid, C are broad spectrum, D all of the above. Should we take a look at them real quick? Here's what sulfa drugs. Oh, that's the wrong lecture. Did I totally close the other? Oh, there it is. Okay. Here's what sulfa drugs do. Sulfa drugs inhibit the synthesis of folic acid, which has a number of different functions, but one of its functions is it's a precursor for nucleic acids and nucleotides. So the sulfonamide, in this case sulfamethoxazole, actually binds to the active site of the enzyme, competing with what's known as PABA for the active site of the enzyme, making it what? What kind of inhibitor? Competitive inhibitor. So it competes for the active site, blocks the synthesis of folic acid. It is a broad spectrum antibiotic. Therefore, it targets not only gram-positives but gram-negatives as well. Oftentimes, if there's one drug, one antibiotic that folks are allergic to, it's this one, or this class of antibiotics. Okay. So to go back to that question real quick then, broad spectrum, blocks synthesis of folic acid, and it's a competitive inhibitor. So for this one, it would be all of the above. Okay. All right, that's it. That's kind of the last of what I wanted to make sure we covered with the antibiotics. At this point, we should have touched on every single one that's on that list in some way, shape, or form, either in a question up on Socrative or in a follow-up question. So I want to just stop real quick and see anything else that needs to be covered with this that folks have questions about or concerns. Okay, let's move ahead now. And what I want to move into today was, in part, what the homework was on for last night. And that's, how do we measure whether an antibiotic actually works or not? Okay? So what methods exist to help us determine activity of antibiotics? This question goes out to anyone. Sitch. What's that? E-test is definitely one of them. What else? Disc diffusion, and I would group that with e-test because it plays off of very similar chemistry in some ways, or I guess methodology. What's the other one? Yeah, broth dilution test. So what I was trying to accomplish last night with the homework, and admittedly, as I've already said, I'm not on my game right now, so I kind of failed in it. I should have an extra one. Is there an extra one? Yay! All right. Oh, I got one over here. Okay, perfect. What I wanted to do was to actually have us practice. This was supposed to be the size of the image you saw on your computer screen, but obviously I didn't realize that computers could do things like zoom in and out and other cool fancy things like that. So the zone of inhibition that you measured was dependent on how big the image looked on your screen. So I printed out these handy dandy wonderful paper rulers so that you guys can practice. Okay? So what we are going to do, I want you just to work on the front side of this right now. I want you to do two things. Measure the zone of inhibition. That's one. And then the second thing is, based on the table below, and I'll talk about what that means, go ahead and determine, is that specific bacterium considered to be sensitive or resistant to those different antibiotics?
I wear glasses. I can't do it.
I do too, but you can do it. It's, all the fun kids are measuring zones of inhibition. Couple of things. One, they combine this idea of the Kirby-Bauer assay, but what else? What else does this potential test tell you? It tells you the minimal inhibitory concentration. So take a look at this. This here is tobramycin, an E-disc of tobramycin, and we've got a gradient. At the top we have 256 micrograms per mil of the drug. Down toward the bottom, 0.016 micrograms per mil of the drug. Basically, we've got a dilution series of the drug along this strip. And the minimal inhibitory concentration is the point at which the organism grows to the disc. So here, 0.064 micrograms per mil of tobramycin we'd consider to be the minimal inhibitory concentration. Okay. What do we mean by that term? What does minimal inhibitory concentration by definition mean? Zach? Not just to have an effect, but actually to completely inhibit the growth of the organism. So some drugs will have an effect. For example, let me flip ahead a slide. We can see with like sulfamethoxazole, there's an effect even after, and the way that I think of it, this is 16 micrograms per mil of drug, 8, 4, 2, and 1. So increasing from 1 to 2 micrograms per mil of drug, there's an effect, but it doesn't inhibit growth. So the MIC is the concentration at which we see growth completely being inhibited for the microorganism. Go ahead.
So if you go to the doctor and like see
Yeah.
like that,
Azithromycin, yep.
how do they determine the concentration of the drug?
Ooh, typically it's based off of weight and age are usually the two. Because a lot of that kind of stuff gets determined in clinical trials to determine how effective is the drug, how much do you need to have. They'll put individuals on different doses to determine how much do you need to inhibit the growth of the microorganism. And that's going to be based on factors such as weight and metabolism. Certain drugs get metabolized quicker than other ones, and so that also can determine the concentration that needs to be provided. Also, the organism's sensitivity.
Because that's like something you take too much, like, or too high of a concentration, really bad for you.
It could be. It could be. That concept there, antibiotics, antibiotic resistance, and the effect on the microbiome, we'll come back to that in about 3 or 4 weeks. Okay, I promise you. It's one, in my mind, it's one of the most important topics probably to get out of this class. Okay. Sydney, did you have a question? Or Emma?
So it's the minimal amount of a drug completely inhibit growth.
To completely inhibit growth. So for example, let's take a look at this, at ethanbutol here. Again, 16, 8, 4, 2, and 1 microgram per mil of drug. At least in this series, what is the minimal inhibitory concentration? 4, right? 16, 8, 4 right here. 2 is too little, the organism grows. 4 is the first point at which we see no growth. Kanamycin, same thing, 4. Jasmine.
Why is it not the one all the way to the left?
Why is it not this one all the way over here? Because, again, the term is minimal inhibitory concentration, minimal amount of drug that's needed to inhibit. So 16 definitely does. Is it the minimal amount? No, 4 is. That's why.
How do you determine the numbers on them? Because in the
I made it up.
okay.
It would be listed for you. Yeah, I fudged data probably. No. So on actually the back side of this worksheet, it lists the first one and it lists the 4. And most of these experiments are done in 1 to 2 dilutions. So if you start with 4, just keep dividing by 2. 2, 1, half, 0.25, so on and so forth. How that gets decided, though, in terms of what's the highest concentration we use, I'll be honest, I don't completely know. I usually like to work in powers of 2 on this, so 2, 4, 6, 8, 6, or 2, 4, 8, 16, 32, so on and so forth. Yeah, you could, and typically it's done in 2-fold dilutions, though. Yeah. Was there another question? I'm sorry, I interrupted somebody, I thought. Does that help to answer that? So on an exam or in the real world, you would know these numbers ahead of time. Those ones were 4, 2, 1, and half, and 0.
What decides which
Left to right or right to left? No, it doesn't matter. Usually in one direction or another, it's increasing concentration. Again, Paige, that would be probably something I would define for you on the exam. But it should make some sense in some ways too. Like even without, you see that this one, there's an increase in growth from left to right. That must mean that the amount of drug is decreasing in this direction. So there should be some intuition to it as well. In some cases, here, the drugs don't work. At least doxycycline over this range has no effect. Some work really well. Streptomycin in this case, we know that the inhibitory concentration is either equal or less than this value. And in other cases, we see a result like we do with the sulfa drugs. Growth, growth, little less, pinprick of growth. And sometimes what experimenters will do is define the minimal inhibitory concentration as the concentration that inhibits 90% of the growth. So if we were to use a definition like that, this right here would probably be the minimal inhibitory concentration. And that's one of the reasons why I encourage you guys to read the methods on papers, is because you have to pay attention to that kind of stuff sometimes. Any questions? Jasmine?
Can you answer question 3?
We'll go over it on... I will, but we're going to go over it on Friday as well. On the back side, you mean, right? So what is the actual MIC for each of those antibiotics? So for antibiotic X, what is the minimal inhibitory concentration? 4 for Y? 2. Okay? All right, so let me just recap in 2 minutes the major points for today. 1, know those general classes of antibiotics and what they target. Okay? Those are ones that I think are most common, and they also serve as great models for studying resistance, and something we'll talk about on Friday. Secondly, ways to measure and being able to interpret these results. That's probably one of the bigger things out of this particular section of lessons. Now, on Friday, what my plan was to watch a TED Talk outside of class, and then for us to have a discussion on antibiotic resistance. There's too many other things that I think I want to still cover before we get to that. So what I want to do is take that lecture and we're going to move it till well after the exam. We'll actually talk about it after we've discussed molecular biology and mutations. So what does that mean? No homework for Friday? Spend the time studying, spend the time with the material so that you come with questions on Friday. I'm hoping to have a little bit of extra time for us to answer some remaining questions, okay?
Is there still a mini lecture for Friday?
No, that means there's no mini lecture for Friday. What we didn't cover today were the modes of resistance. If you want to prep for Friday, that's what you should be reviewing. So go back to mini lecture 2 for this section and just review the second half of it. No homework. I'll take them if you want, otherwise keep them. Enjoy playing with rulers, paper rulers. All right, have a good afternoon.
What time?